Good day, everyone. Today we have a really exciting interview for you. The interview guest is Colleen Cutcliffe, and she's the co-founder and chief executive officer at Pendulum. She has more than 25 years of experience leading and managing biology teams in academia, pharmaceuticals, and biotech. Prior to starting Pendulum, Colleen was the senior manager of biology at Pacific Biosciences and a scientist at Elon Pharmaceuticals. Colleen completed her postdoctoral studies at Northwestern Children's Memorial Hospital, and Colleen received her PhD in biochemistry and molecular biology from Johns Hopkins University and her BA in biochemistry from Wellesley College. Hope you enjoy the interview today. Let's get to it. Well, thank you. Firstly, thank you for taking your time to speak with us. I'm super excited, and as I mentioned earlier off air, part of my background was listening to your interviews kind of at the start when you launched your flagship product. I think you spoke to Dr. Gundry, and then there was a follow with Dr. Hyman, Dr. Mayer, and all of that, just really fascinating stuff. And could you start with sort of a short intro to Pendulum and what got you going there?
Sure. Well, I think this was a little bit over 10 years ago now. We were starting to just begin to unearth what the microbiome is. And actually, when we were first fundraising, I literally had to explain what the microbiome was. You have all these microbes inside and on you, and nobody knew what it was at that time, which is funny because you fast forward to today, and I feel like every investor certainly knows what the microbiome is, but it's just really exploded. So at that time, it was really an early science and primarily in the academic realm. And what we realized was that there was an opportunity to use DNA sequencing technologies and traditional kind of pharmaceutical methods of creating products, but target the microbiome, where you could have a vision to create a family of products that had the efficacy of a drug, but the safety of a probiotic. And when we came into this, myself and two co-founders were all very technical in background. And so we were really coming at it from a technical approach of mapping the microbiome and figuring out where there's opportunity for next generation probiotics, essentially. And then on a personal note, as I started learning more about the microbiome, I realized that I myself had something to gain by learning more about it. And it came through my first daughter. So she was born almost 2 months prematurely. When babies are born that early, they give them multiple doses of antibiotics. And she was in intensive care for a month receiving these antibiotics, not because she had an infection, but because it was prophylactic to keep them from getting an infection because they're so fragile. And as she got older, I started to realize that she had food sensitivities that the rest of us in the family didn't have. She was the kid at Baskin-Robbins asking how much dairy is in the sherbet. And she, has always eaten a lot of salads because that worked better for her. And this paper came out in 2012 where Dr. Blazer showed that when you looked at infants who were systematically on antibiotics, as they got older, they became more prone to things like obesity and type 2 diabetes than other people. And that study was actually repeated by the Mayo Clinic a few years ago. And they showed that kids who were 2 years and under, who were on a lot of antibiotics, were more prone not just to obesity and type 2 diabetes, but also things like celiac disease, allergies, asthma, ADHD. And so this early start to life where you're completely decimating someone's microbiome sets them on a path where they're depleted and sets them on a path for chronic illnesses that show up, decades later in life. And I think a lot of people have had this experience of going on antibiotics, having GI distress. Some people on the other side of antibiotics really notice health changes. And so our mission was really, how can we help millions of people get a healthier microbiome to overcome some of these things that we do? And for me, I felt like we could help millions of people, including my own daughter. So off we went to start a company.
Amazing. So you started with glucose control. And so why did you start there?
We started with glucose control because we ourselves did some studies in twins where we were looking for discordant twins, where one twin had diabetes or was obese and the other twin was ostensibly healthy. And we were looking at the differences in their microbiomes because they're genetically identical. And so we thought that might help clarify, what was the microbiome component that was playing a factor here in their metabolism. In the meantime, a lot of other people around the world were also exploring this. And so there was a lot of evidence to suggest that people who have metabolic syndrome, obesity, prediabetes, and type 2 diabetes are low or entirely missing certain functions in their microbiome. And that specifically those functions are strains that help you metabolize fiber into short chain fatty acids, including butyrate. And butyrate is known to be an upstream modulator of GLP-1, which is known to modulate insulin and glucose. And so there was this very clear mechanism of action there. And we all know a high fiber diet is really good for us. You can actually make claims around helping metabolism with a high fiber diet. We all know insulin and GLP-1 are highly effective for people with type 2 diabetes. But what was missing was this component, which is the microbiome that actually does that metabolism of fiber to GLP-1. And so that became solid gold for us. Let's go after, how do we isolate those strains? How do we grow them up? How do we start delivering them and see whether they can provide benefit?
So this product, as I understand it, is multiple strains, right?
It is. It's a consortium of strains, and actually it also has a prebiotic fiber in it. And these are actually function together. So the breakdown of fiber into butyrate is actually a multi-step pathway. And so what's included in here are all the strains required for that full breakdown.
The should correct me if I'm wrong, but it seems like, later when you released Akkermansia on its own, that there was some sort of like, this is the shining star that needs to come out a little bit more. What made that unique? Because that was in that original blend, right?
Yes. Well, I'll let you we did not come up with that on our own, the releasing Akkermansia by itself. What actually happened was we had customers saying to us, hey, I really love Akkermansia. I'm buying this big formulation, but all I really want is that one strain. And would you just sell that? And then we had functional medicine and integrative medicine doctors asking us, would you just sell Akkermansia? That's all I want to give to my patient. And then there were, Reddit threads about it. And so we said, all right. This could be a very small number of people, but it could be something here. So why don't we just make 1,000 bottles of just Akkermansia and see what happens? Well, Jonathan, we made 1,000 bottles, and in less than 10 days, every bottle was gone. And so we thought, holy smokes, there really is a market out here for this strain that I thought nobody had heard of. And so we then proceeded to be in and out of back order for the next 9 months, because every time we made a new batch, they all got sold. And I think the thing we didn't necessarily expect either was that the retention was so high. So people were returning back because they were experiencing all sorts of different benefits. And we're only now starting to get an understanding of what are all the different benefits of Akkermansia that people are experiencing, and it's been fascinating.
Let's dive into this bacteria a little bit more, if because there may be some people who know what this is, and I have this kind of bookmark question in the background, which is how the awareness, what caused you think the awareness of Akkermansia to even be there for the request to make of your company, but also tell us some of the properties, what it does in the gut as well.
Yeah, so Akkermansia is the only strain that we know of that literally lives in your gut lining. And its job is to make sure that that gut lining is regulated properly. So I think about it like I have a wooden fence in my backyard. When I first moved in, that wooden fence, which has all these planks, was really strong and shiny and brand new. But over time, through seasons and wear and tear, the glue between those planks can start to weaken, and a plank could fall. And your gut lining is literally the exact same way. You have these planks that are lined up and held together by glue, and Akkermansia's job is just to sit at that fence all day long and make sure that if anything's getting weak, it's fixing it and mending it and keeping that fence strong. And what happens over time is that you start to lose Akkermansia. So what we know is that people with obesity and diabetes are low or entirely missing Akkermansia. We know that people with inflammatory issues are low or entirely missing Akkermansia. We know that as you age, you become depleted in Akkermansia. We know that when you go through periods of high stress, you start to lose Akkermansia. We know for women, when we go through menopause, we start to lose Akkermansia. And so there's a lot of reasons why you have this strain and you start to become depleted over time. And now that gut lining and that fence is not being properly regulated. And the outcomes of that are just extremely vast.
Is that glue that you're referring to the mucin layer? Is that the glue? Okay, if you
Exactly.
explain, because I think people generally have the understanding, at least if they've been paying attention to sort of the functional medicine realm, that these tight junctions, like the separation of these tight junctions, is part of the issue. But I'm not sure that people understand that there is that glue there That is, there's a buffer zone between the tight junction itself and everything else that's in the gut.
Exactly right. You have the planks are literally your cells, your epithelial cells, and these tight junctions between them, they are held together by this what I call glue, which is mucin. And mucin is something that your microbiome actually produces. And so Akkermansia actually has two roles. One is that it can break down that mucin when the mucin is getting insufficient, and the other thing is that it can rebuild that mucin. And so there's two parts of this. So it's, before you go paint something, you strip it down, remove all the old paint, and then you put new paint on, and it's literally doing those functions. So it removes it can consume the old mucin, and it can produce, along with other strains in the microbiome, it can produce mucin. And so that is the key part of maintaining this strong gut lining.
How does it get there? Is it part of the sort of the mother's lineage of passing down the microbiome, or is this something that, how does it get there? Do you have an understanding of that?
Oh man, such a great question. How do any of those microbes get there? I think initially there were reports saying when you're born, you are a clean slate. There are no microbes. And so the first microbes you get are if you're born vaginally, through just passing through that birth canal. And then there was this idea of, oh, if you had C-section babies, they don't get that advantage. And so I know people who took, vaginal microbiome and tried to smear it over their C-section baby in order to try to seed good microbes. We do know that some of the early microbes in infants include Bifidobacterium infantis. But over time, how you get these other strains is not exactly clear. Because, for example, Akkermansia, there has never been a report of Akkermansia on a single food. It doesn't appear to be a strain that naturally becomes a spore. So some strains, like Clostridial strains, they go into a form called a spore, and then that's kind of in the air and all around us, and that's probably how they get into our bodies. So it's not clear. It's definitely, we're not born with Akkermansia in us. So we're getting it from somewhere. It could be from the mother. It could be from breast milk. There have been some reports that there's Akkermansia found in mother's milk. It could be through different foods. In the environment, we actually don't know how you get it.
You listed these different scenarios in which Akkermansia starts to decline, aging being one. Uh, what's your understanding of what causes that, the decline of Akkermansia?
I think that when we think about things like diet, it becomes a little bit clearer because your diet is not just food for you, but also food for your microbes. And so if you have a diet that isn't high in fiber or high enough in fiber, or you don't get polyphenols into your diet, those can be reasons why you lose Akkermansia, because you're just simply not feeding it. And so other strains start to pop up. It's an ecosystem of competition. But I think it's not entirely clear what has led to this correlation of losing Akkermansia and why people start to lose it. But I think certainly what's clear is that it's probably got to do with the ecosystem of microbes there and what else is kind of going up and down. And then the part that is really unknown is how do we, the host, affect and impact our microbiome? And we look a lot at how our microbiome impacts us, but what are we doing to shape the microbiome? That's not totally clear.
Yeah, I mean, what part of my, as I mentioned, we have this retreat coming up, and so I've been just doing a lot of digging around it, understanding what you just explained, our impact, and it seems like our mood, our stress levels, these hormones and neurotransmitters are sort of changing the composition of the microbiome. Is that a component, and how much of a component do you think that is on Akkermansia and the other good microbes?
I think that it's emerging as a bigger and bigger component. It's just a lot more complex to understand. But I think the fact that, when you're undergoing periods of high stress, we know that there's, significant hormonal changes. Obviously, menopause is defined by hormonal changes. And so I think that what we're starting to see is that there is an interlinking here, and that if you can use different tools to try to maybe mitigate some of the hormonal changes or maybe the swings, that might help. But, this is a super early science. So I don't think that I have seen enough evidence to say for certain this is how one would do it.
Okay, so you launched glucose control, then Akkermansia followed that. Can you take us more through the Pendulum story of the other products that you launched and where you're at now?
Sure. So we launched Akkermansia as a single strain. We then also launched Clostridium butyricum as a single strain. And this was, again, this is a strain that's actually been sold in Japan for decades, and it's been used for treating IBD as well as IBS, so Crohn's disease, ulcerative colitis, and those flare-ups that are associated with it. And the issue with the product in Japan is that the dose is so low that you literally have to take 30 pills a day in order to get the effective dose. And so at our company, we have R&D. We also have manufacturing. These are all strict anaerobes, and so they're actually quite tricky to manufacture. We hadn't planned to manufacture them ourselves. We had tried to outsource that, but all the other probiotics on the shelves right now don't have this requirement that there can be no oxygen in the entire manufacturing process. And so you literally from end to end have to keep oxygen out, which is quite challenging because of course oxygen is everywhere around us. So we figured out how to manufacture these strict anaerobes, and that enabled us to create butyricum at a much higher dose. So now what you can get in 30 tablets elsewhere, you can now get 10x that in actually one tablet. And so we released that, and it's been primarily used for GI repair and gut issues. And then our most recent product that we released a couple of months ago is Metabolic Daily. And this is sort of an answer to a lot of the kind of critique that we got on pendulum glucose control. So pendulum glucose control, published in BMJ, clinically shown in a placebo-controlled, double-blinded randomized trial to lower A1C and blood glucose spikes for people with type 2 diabetes. Just really, I think, the first and only probiotic that is really showing efficacy at a clinical level for people with diabetes. Well, the critical feedback we got on that was, hey, what if I want to reduce my blood sugar spikes, but I don't have type 2 diabetes? Or, what if I don't have a high A1C and I don't have these really high glucose spikes? Is that going to harm me? Also, I don't have $165 a month to shell out for this product. And we didn't price it there because we were making a killing off of it. We priced it there because it's actually expensive to make. And so what we did was we created Metabolic Daily. It is exactly the same formulation as pendulum glucose control, except it is at a lower dose. And so this is really for anybody who wants to help their bodies metabolize carbs and sugars better. And it's at a $49 price point, which is much more affordable than $165. And that lower dose enabled us to then bring it down to that price point. And so that is sort of the arc of the different products we've released. And I should say along the way in December, we also released an add-on product. So when you buy any of these products, you can also add on polyphenols. And the polyphenols were selected based on work that we did with our Akkermansia strain to try to pick polyphenols that actually work well with our Akkermansia. But polyphenols have been clinically shown by lots of different groups to increase your Akkermansia levels and have a variety of health benefits. And so this is really paired with products in order to kind of give yourself the 1-2 punch of getting more Akkermansia in your body.
Okay, so the Akkermansia is partially eating the mucin layer, but it is also eating polyphenols. Is that correct? Is that it's got multiple food sources, and what are you feeding it in the lab to get it to grow?
It does have multiple food sources, glucose, being one of them, sugar being one of them. And actually one of the challenges to growing Akkermansia in particular is that it feeds off mucin, and mucin gets created, it's sort of a meat byproduct. And so when you grow this strain in the lab, and I should say this, in the United States, you're not allowed to grow probiotics in any sort of meat-based media, which just as an interesting side note, was a rule that got made when we had mad cow disease come through the world. And so because of mad cow disease, they made this rule like, oh, you can't grow probiotics in any sort of beef broth. And so these strains that we grow are vegetarians. We have to figure out like a vegetable-based media to be able to grow them in. And so for Akkermansia, that is challenging. And so we figured out a variety of different energy sources that we can use in order to grow the strain. The polyphenols are actually really interesting because I don't think we fully understand how the polyphenols are resulting in increased Akkermansia, because actually polyphenols are not one of the prebiotics that feed Akkermansia. That's not totally clear. And so the current and furthermore, polyphenols have almost like a mild antibiotic effect. They appear to kill certain strains. And so the current leading hypothesis is that the reason polyphenols are beneficial is because they actually get rid of some of the less beneficial strains, and that enables the ones that you want to really thrive. So it's just another indication of how complex the ecosystem is.
So polyphenols being the sort of plant constituents that provide pigment to different plants, and the examples of this are green tea or pomegranate or blueberries or that sort of thing.
Absolutely. Or if you like, red wine, chocolate. But
Right.
yeah, the polyphenols that we found really worked well with our Akkermansia were derived from green tea, exactly, and pomegranates.
Amazing. So how does one, knowing that you have all of these different products, how does one go about starting um, to choose one? And is it an experimentation process, or is this sort of doing a microbiome test to see what one might be low in, and that's what you would buy to supplement? How do you go about that?
I think there's a couple of ways to tackle it. The first is there a specific problem that you're trying to solve? And so if you're having a particular GI issue, or I really want to manage my blood glucose spikes, that can help you figure out, what's the right product for you. We actually have a quiz on our website that helps people hone in on, what are you trying, what is your intention, what are you trying to get done, and we can guide you to the right product in that way. Alternatively, people can take a microbiome test, and there are certainly individuals and physicians that use microbiome testing to see what strains their patients are low in. And this is where a lot of people kind of hone in on, oh, I'm low in Akkermansia, I just want to get Akkermansia back. And I think that is a legit way to make that decision. I would just sort of say that we are still in the very early stages of this science. And so being low in one strain and just replenishing that strain may work fantastically, but there may be other things that we don't know about that are codependents, that you may also want to boost. And I think we're just at an early stage for not fully understanding that. So I think if you've taken a microbiome test, you want to increase your Akkermansia levels. Akkermansia is absolutely the thing. I think if you have a very acute set of GI issues, you could start with either Akkermansia or butyricum as a single strain. It might also be that many people don't want to start with a full formulation. They like to, simplicity is the key. And so if you kind of feel that way about it, I think it makes sense to sort of start with a single strain and then build from there. Alternatively, and I kind of fall into this camp, I would start with the full formulation. If that worked for me, I then might start to cut things out and say, okay, what is the bare minimum that I need to see to hold on to that efficacy? So it really is sort of a personal choice, and a choice that people can make with their physicians to decide how to tackle.
What about mood? Of the strains that you produce, what do you know about their effects on mood?
Well, I think there are a lot of publications on the role of short chain fatty acids and butyrate on mood and how those things interact with each other. With Akkermansia, we've actually kind of really honed in on trying to understand it, because we got back 70% of our customers were telling us that they have reduced sugar cravings when they're on Akkermansia or glucose control, which has Akkermansia in it. And we're starting to hear people say that about Metabolic Daily. It's only been on the market for 60 days. So we're just now starting to get information about what people are experiencing. But this reduced sugar cravings and reduced food cravings is particularly interesting. We know that Akkermansia is an upstream modulator of GLP-1. So Akkermansia, increases GLP-1. And we know that GLP-1s can help you with weight loss through satiety. So you just don't feel hungry anymore. And I think that when you're not craving foods and you're not constantly fighting that battle of, oh, I really want that thing, but I'm going to restrict myself from having it, or I really want that thing, and so I ate it and now I feel really bad about it. There's sort of a very negative mood cycle that you can get into that is driven by your cravings. And so being able to help people reduce those cravings, I think, is an important part of being starting on the good hamster wheel of mood. And then separately, or maybe connected, we don't know yet, to that, is that Akkermansia can also produce GABA. We know that the gut-brain connection is a really important part of mood. We know that there's this vagus nerve where neurotransmitters can actually go from your gut to your brain, and we know that the gut produces a massive amount of serotonin and GABA and dopamine. And so, why is it doing that? What is it doing? I think that's still being uncovered, but we know that Akkermansia produces lots of GABA, and we also know that we have customers reporting reduced stress and anxiety, better sleep, less kind of brain fog as they go through the day. And so I think when we think about mood, there's sort of the calming and then the reduced food cravings, which may be connected to each other, but it's definitely in the early stages, but an interesting part of what your gut is doing.
I can't recall if you shared the story with me as we were setting up, or you did when we started recording, but you were saying at one point that you found a lot of GABA in sort of the growth medium when you were growing Akkermansia. And is that something that could be ingested? Could you ingest that GABA that has been in that medium and get the benefit?
Oh, now you are hitting on something that we debate heavily in this company. Can we all just do shots of that media? Well, it's actually a really recent finding for us. I mean, we've only known about this for a short period of time, and it was because we knew that we knew that Akkermansia was capable of producing GABA, and didn't think to necessarily look in the media. And as you said, when we look into the media that the cells are growing in, which is literally what we throw away after we grow the cells, we found massive amounts of GABA and actually a bunch of other small molecules, which we'll save for another time to talk about. But we are very interested in one of two ways. One is, could you just consume that liquid media and have health benefits? And the other is, can you then almost like dehydrate the thing? So get rid of the media and get it down to just the small molecules and then ingest that. I myself am happy to do shots of media, and I just need to convince the manufacturing team to give me some.
Sign me up. I'll be your lab rat.
All right. Actually, I might take you up on that.
uh, what, where do you think the microbiome science industry is going? What do you see as the next stages?
I think there are a fair number of companies in the pharmaceutical space that are really thinking about how do we create microbiome interventions that, as we know, target C. difficile infection, that can target IBD and IBS, and some of, especially with IBD kind of going after inflammation. And I think we're seeing some good work being done on the gut-brain connection, and particularly I'm really fascinated by the work that's being done in Parkinson's and Alzheimer's through the gut microbiome. Actually, when I started my career, it was in pharma, so I kind of have started on that side of the table and made my way to the natural side of the table. We were trying to create drugs for Parkinson's disease. And one thing that was really fascinating about that is that when we were doing drug discovery for Parkinson's, we were so focused on the brain and these plaques that show up in the brain in both Parkinson's and Alzheimer's. And oh man, how do we get rid of those plaques? And I think what we're starting to understand is that not only do you have neurons in your brain, which once they're dead or once they form these plaques, you're kind of SOL, but you also have neurons in your gut. And unlike the ones in your brain, the neurons in your gut, they turn over. They're constantly refreshing and replenishing. And that these plaques that show up in the brain actually first show up in the gut. And so the new theory is you actually have some problem in your gut microbiome where these neurons are forming these plaques. And then, as we know, they're sending signals to the brain. And then by the time that shows up in your brain neurons, you're already too far gone. So can you go to the earlier stage and target the gut microbiome to make sure that those neurons stay healthy? And you have a chance there because they're constantly turning over. And so you can almost think about, kind of a stem cell equivalent. If you could keep those guys fresh and replenished, maybe you could really protect your brain. I think that is a really interesting place. And on the other end of kind of the neuronal spectrum is autism. And we know that for some kids, when you change their diet, you can actually change their symptoms and improve them significantly. And when you change your diet, one of the main things you're changing is actually your microbiome. And we know that kids with autism have a different microbiome than their healthy siblings. And so you can start to tackle that as well and realize that maybe it's not got to do with the brain, but it actually has to do with the gut.
It's amazing. Well, gosh, Colleen, thank you. Thanks so much for coming on and sharing all that with us. And good luck as you venture forward in your new products and everything else that comes out.
Thank you. Thanks so much for having me, and happy to answer any other questions.